in using molecular bioinformatic and statistical approaches to study biodiversity, as well as design tools for biomonitoring. Dr. Littlefair is currently a lecturer in biological sciences at Queen Mary University of London in the UK. Dr. Littlefair has a strong connection to the Canadian limnology community due to her recent experience as a postdoctoral fellow at McGill University and her eDNA research at the Experimental Lakes area. Our first panelist is Dr. Irene Gregory-Eves. Dr. Gregory-Eves is an associate professor and Canada Research Chair in Freshwater Ecology and Global Change at McGill University. Dr. Gregory-Eves is also a member of the Royal Society of Canada's College of New Scholars, Artists, and Scientists. Central to Dr. Gregory-Eves' research program is the ability to develop, scrutinize, and integrate data from different sources, including lake surveys, time series, and field experiments. The study of natural environmental archives, especially lake sediment records, is also a key part of her research program. And our other panelist is Dr. Michael Rennie, who is an associate professor and Canada Research Chair in Freshwater Ecology and Fisheries at Lakehead University. Dr. Rennie oversees his research team's use of whole lake experiments, lab studies, and archival data sets to investigate how lakes and fish populations respond to ecosystem change, such as climate warming, invasive species, and contaminants. He is also the acting director of Lakehead's Aquatic Toxicology Research Center. Dr. Rennie was recently awarded an inaugural service award from SEL for his years of dedicated service to our society as my predecessor in the role of communications officer Anglophone. And so I would like to give the virtual floor to Dr. Littlefair. Thank you.
Thanks so much, Andrea. That was a really nice introduction. And thank you for inviting me to be your first Limno seminar speaker. So I'm now in a faculty position at Queen Mary University of London in the UK, but the majority of the work that I'm speaking about was actually conducted while I was a postdoctoral fellow at McGill University in collaboration with the Experimental Lakes area. So hopefully that's a connection that's close to your hearts. Just to say, today I have a little cough cold thing, so I'm really sorry if that disrupts the seminar. It's not coronavirus, it's just a cold. Okay, so thinking about environmental DNA. So here is a lake habitat. And the water here contains traces of fish, zooplankton, amphibian, and mollusk DNA, all the DNA of the animals which are living there. So now and in the future, the key parameters that we use to describe an aquatic community like this, like species richness, diversity, evenness, and abundance, might be measured with environmental DNA. So to sample a lake habitat like this, we would take eDNA from the water, filter it, and sequence our targeted amplicons, so to target a particular species of interest. And this is a really growing field. So these, here I've put just a list of companies and governments who are now doing habitat assessments with eDNA right now, including those which are mandated by environmental legislation, such as the EU Habitats Directive. So the way that this works is that we'd be dealing with environmental samples which contain a mixture of different species DNA. These environmental samples could be substrates like water or sediments, or maybe we're looking at gut or fecal contents if we're interested in diet analysis. We isolate the DNA by extraction, and then we have a number of options, different molecular techniques to perform depending on whether we're interested in targeting just one species, or perhaps we're interested in mixed species analysis or meta-barcoding. More recently, even it's possible to sequence even longer fragments of DNA. So that would be sequencing longer parts of the mitochondria or the nuclear genome in a technique known as metagenomics. So we have a number of options depending on the goals of our study. This is a bit more information about how metabarcodes work. So this allows us to discriminate within and between species. So our aim here is to find gene regions which give the best taxonomic discrimination between species and match these to reference databases. So in recent years, there's been a huge effort to determine the best genes in which to do this, and also to build up our reference databases. So we have genes that we can match against, kind of like a library of DNA. So I've explained a bit about what eDNA is, and now thinking about the promises and challenges section of this talk. So I did find, as I was researching this talk, that all the promises and challenges are kind of mixed together. So there's kind of challenge in the promise and promise in the challenge. But these are the sort of 5 main things that I want to mention. So first up, eDNA is being used for biodiversity assessment by a large number of different bodies now, including at government and industry level. So I've got 3 potential applications here. So the first is monitoring rare or protected species. So an example of this right now is in the UK. We are protecting the great crested newt, and that protection is mandated by the EU Habitats Directive. So it's part of our legal biodiversity framework. And the government has now started accepting eDNA assays as being evidence for monitoring the presence or absence of this newt. Also in Europe, we have eDNA assays now to monitor invasion fronts. So this is the signal crayfish in the middle picture. So these are American, but actually they're invasive in Europe. In the US, the United States Geological Survey is actually doing a lot of work on invasive species eDNA assays, in particular with zebra and quagga mussels, so developing those assays so they can see where the mussels have colonized. And finally, eDNA is of use when we're thinking about general biodiversity surveys in multiple areas. So what are the rates of detection like when we think about sampling a lake with environmental DNA? So from my own studies so far, so we're generally getting good species coverage with environmental DNA. So this data is from a 21 lake survey conducted at the experimental lakes area. And here I'm showing sample accumulation curves where each site is a point within a lake, 1 water sample, and each curve is represents a single lake. On the left-hand side, these 3 graphs, we are targeting all metazoa. And on the right-hand side, we're just targeting fish. You can see on the right-hand side, when we're just looking at fish, we actually have really good species richness estimates with environmental DNA. So we detect all the fish known to exist in the experimental lakes area, plus 2 others, which they don't usually record in their ongoing monitoring surveys, but still feasible as being within the area. On the left-hand side, when we're looking at metazoa, we don't get quite as good coverage, but we do see that these groups are more diverse. So we don't get the same plateauing out of the sample accumulation curves, but we still have sort of reasonable estimates of biodiversity. And we have good species level discrimination for these different groups. Thinking more broadly about broad patterns from many different freshwater studies, there's been a lot of discussion about how eDNA compares to conventional methods in terms of detection. And by conventional methods, here I mean nets, traps, and visual surveys. So these are results from a meta-analysis that was conducted by Mary McElroy. And she compared species richness as detected with environmental DNA and conventional methods. So on the x-axis, we have the species richness of the study. And on the y-axis, we have the difference in species richness between eDNA and conventional techniques. So if that number is positive, it means that eDNA detects more species. And if it's negative, conventional methods detect more. You can see that there's a large degree of congruence between the two methods. Perhaps eDNA detects slightly more species. Detection means different things to different people, though. So for some people, presence-absence data is sufficient, depending on the goals of their studies. Some people are interested in repeat detections to ensure that whatever they're seeing is a reliable signal, so sort of similar to occupancy studies. And of course, a burning question is looking at estimates of population biomass or abundance. So there have been several studies now, particularly on single species assays, so just looking at one target. For this, we use a technique called quantitative PCR in freshwater ecosystems. So individual results from different studies do vary, and more work is probably needed to explain why they vary. But there are actually enough studies now that a meta-analysis could be performed. So this meta-analysis in particular was conducted by Matthew Yates, and he looked at 44 different correlation coefficients between eDNA sequence counts and species biomass or density over 19 studies. So he actually found that the correlation between eDNA particle concentration and abundance, organism abundance in laboratory experiments was extremely strong, explaining up to 81% of variation observed when it's converted to the R-square value. And although it's a bit weaker, there's still correlation between eDNA counts and species biomass or abundance in natural environments as well. So there are fewer studies that feature mixed species results, although this is actually something that I've also examined in collaboration with the experimental lakes area. So this is a study on that same survey data, which looked at eDNA counts and compared it to the abundance categories that the experimental lakes area place their fish in. So when they go and do their long-term biomonitoring surveys, they actually classify the fish as being rare, moderate, or abundant. So we don't yet have estimates of how well eDNA would be explained by biomass estimates in mixed species samples, but often rank-based results or results featuring relative abundance can be quite informative. And it looks like there is a good correlation between these two things. Thinking about the temporal scales of detection, so how long eDNA persists in the environment. So this is an interesting question because we want to make sure that eDNA persists for long enough such that we actually have time to detect the organisms that we're interested in. We don't want too much degradation. But at the same time, if eDNA persists for a really, really long time, we have that, you can almost think of them like sort of smoke signals of animals' presence in the environment. So if they persist for a really long time, then we might be detecting populations which are no longer present in that environment. So we kind of want a balance between the two. So lots of these studies have been on animals in sort of mesocosms or caged in sort of enclosures within lakes. So this was a study from Barnes et al. In 2014 featuring the common carp in mesocosms, and they found that detection probability did dramatically decrease after a few days and generally didn't persist for more than a few weeks. I think, though, that there needs to be more examination of these factors in natural environments, because when you have animals in cages or in mesocosms, generally they behave in different ways and they release DNA in different ways to when they're sort of free swimming in the environment. We do know, however, that some DNA settles from lakes and actually gets trapped in sediments. It's bound to sediment particles, and several people have started to use sediment cores as a way to examine long-term biodiversity changes in lakes. So that's actually something that I've begun exploring this year in collaboration with Renee Gregory-Eves, who's going to be one of the panelists in this event. So once it enters the sediments, DNA, when it's bound to those particles, does become very stable, and thus might be a signal of long-term biodiversity change once it's stabilized. Finally, just the last part of my talk, I wanted to talk about different spatial scales of eDNA detection. So to some extent, this is influenced by its kind of temporal range as well. I suppose once DNA persists in the environment, it can then be transported on different spatial scales. So a key difference between conventional methods and using eDNA for biodiversity survey is that we have to determine what those molecular signals mean in light of water movements and other environmental forces which might be moving those signals. And also the high degree of connectivity between aquatic habitats. So as I said before, we can sort of think of molecules as a smoke signal indicating organisms' presence. So normally in lakes, animals prefer to use different parts of habitat. So here is a cartoon with different fish species in different lakes at the experimental lakes area, as well as, as well as fish using different zones of the lake, so pelagic and littoral zones. So if we imagine that animals are characterized by their habitat niches within lakes, does the eDNA that they release stay within these zones? Or does water mixing mean that the different samples taken from this habitat are genetically homogeneous? So where is the best place to sample in order to get a representative picture of biodiversity? It may also be that there are even wider catchment signals in lakes. So this has been studied in more depth in river tributaries. And authors have found that in some places, rivers can actually act as conveyor belts of biodiversity in terms of collecting signals from different areas in the catchment. So this will all affect how we design our sampling strategies for organism detection with eDNA. And there's actually very little work on this, despite the ubiquity of eDNA work being done on lakes. So very few people have actually looked at where the best places to pick our samples are in lakes based on our understanding of the spatial temporal signal of DNA molecules. So these studies took place at the experimental lakes area in Ontario. And I actually studied, for this particular analysis, 23 lakes in 3 connected lake chains, which you can see coloured on the diagram here. So many of these lakes have a distinct community assemblage, which is how we can validate which molecular signals belong to which lake. And ELA have been monitoring their lakes since 1968, which is absolutely brilliant for me because I can validate my molecular results with historical records. So we sampled in all of these lakes shown, and we sampled each lake multiple times in different areas. So the pelagic, littoral and profundal zones to pick up different fish and zooplankton species. So after sequencing, we matched all of our DNA sequences to reference libraries and identified the different animals. So this is kind of a complicated graph, but these are results for within lake variation in eDNA signals according to sample position. So on the x-axis here we have the position of the samples, so whether the water sample was taken in the hyperlimnion, the shoreline, or a transect across the middle, which capture more pelagic species. And the colours represent the habitat classifications of different species. So where those species usually like to live. So what I want you to take away from this is the strong interaction between how species use habitat and where the samples were taken in predicting the DNA sequence counts. So for example, on the left-hand side, we have we're thinking about just the fish. And it seems that while littoral and mid-water benthic fish can be detected in most places in the lakes, hyperlimnion dwelling fish, profundal fish, can only really be detected in the profundal zone. And it's a similar case for zooplankton. So those profundal zooplankton can only really be detected at the bottom of lakes. So that's a really useful thing to know if we are interested in targeting those particular organisms. And that's something that so far people have not integrated in their design of eDNA surveys in lakes. And without that information, we'd actually be undersampling species richness. Again, in collaboration with Mike Rennie, who's one of the panelists here, I've been looking at this question of within lake spatial variation and detection in more detail. So this is a separate study in which we looked in more detail at vertical eDNA distributions. So taking, oh, I'm sorry, I think that automatically moved forward. So taking a more nuanced approach to looking at warm water fish and those which are using profundal habitats because they're cold water fish. So here we sampled 7 lakes at depth gradients between shallow and deep water at the center point of each lake. So kind of like a depth profile cutting the lake. We also wanted to contrast the seasonal effects of lakes during stratification and turnover. So during stratification, there's relatively little water mixing due to the presence of the thermocline. And that could be explaining these strong differences in signals between the top and the bottom of lakes. As well as interacting with the fish's habitat preferences. So it looks like, so here we have results from those studies. So here are different proportions of fish species detected at shallow and deep parts of the lake. So these top bars are the most shallow points of the lake, and the bottom bars are the deepest. And we have results from stratification and turnover. And it looks like eDNA becomes very strongly stratified either above or below the thermocline during the summer, in a way that reflects the thermal niches of fish. So for example, very few cold water fish species can be detected at the surface. Relatively few sequence counts are present there. Lake trout can't be detected at all if we just take surface samples. And that's problematic because often we're quite interested in game fish like these, and also as important predators in freshwater ecosystems. In fact, warm water fish DNA is much more prevalent at the surface and much less prevalent at the deeper depths. During turnover, however, water mixing ensures that these signals are homogeneous, so that there may also be some contributions from fish habitat selection as well, because cold water fish at this time can use these surface waters. So these results overall are suggestive that when we get much better community representation when we sample during the autumn, or when we include samples from both shallow and deep depths. So it really matters in a lake where you sample, even if those lakes are relatively small, like the ones at the experimental lakes area. And by integrating all this knowledge, we can really make sure that eDNA is a reliable technique for estimating species richness of all the different species living in the lake. Finally, I just wanted to zoom out again and think about when habitats are connected. So thinking more about how signals might be flowing between habitats. So when lakes are connected by small streams. So on the left here, we just have a reminder of what that might look like. So these, I selected 3 lake chains at the experimental lakes area to study and survey. And I was observing the downstream journey of DNA and how that is shaped by, excuse me, how that is shaped by the rate of flow between the different lakes. So we do find that so we classified eDNA hits in this survey according to whether they were expected or unexpected based on fish surveys and the historical data from the experimental lakes area. So we do find that as we progress downstream in these lake chains, or we look at lakes with more connectedness, so more lakes connected to them, more unexpected detections accumulate. So this is a pattern which does not appear with expected detections, and the pattern is also consistent across the 3 networks that we examined. So we kind of have a sort of tension here. On the one hand, eDNA does appear to reflect quite local differences in species richness when we're considering, for example, those differences between the epilimnion and the hyperlimnion species composition. But on the other hand, a small amount of signal is being transported downstream, which can be problematic when we want to just represent the molecular signals from one particular habitat. So just to summarize, so I really feel that species detection with environmental DNA is really promising in terms of the molecular and bioinformatic tools. So generally, recovery from mesocosms and mock communities in the labs is really, really strong. And it's also improving dramatically in the field as well, which is really where we want to see those gains, because ultimately we want to apply this as a tool that can help us in conservation and habitat management settings. The tools are somewhat taxon dependent, so there's a strong focus on fish in my research, for which I can only apologize. But you know, if you were looking at macroinvertebrates, there would also be quite adequate taxonomic coverage in terms of reference libraries. Just other taxonomic groups sometimes can be a bit difficult. For relative abundance, eDNA does show promise, especially when we're looking at single species analyses, or we have questions that can be answered with rank-based abundance. So saying species X is more common than species Y in this habitat. The temporal scale of eDNA detection is likely to be quite sensible and suits our purposes if it's just limited to DNA molecule persistence from a few days to weeks. But spatial scales of DNA and the transport of DNA around aquatic systems definitely need more examination, and that's going to be really important when we're thinking about optimum sampling designs. So therein ends sort of my thoughts on promises and challenges. I'm really excited to take your questions. I'd just like to thank the enormous team of people who are helping and collaborating with me on all of these things, especially my field assistants, without which none of this is possible. And thank you very much for your attention.
Just imagine you're hearing this virtual applause. Thank you so much.
You.
Oh, there we go. There's
I can see an emoticon.
That's great. Well, thank you so much.